Yeah. So good afternoon, ladies and gentlemen. And we are going to continue our lectures. And today we're going to take classification of amino acid. I believe before now we have done the introduction class, and we have been able to tell us our expectation. But what are the things that you're expected to know after this class? After this class, you are expected to know how to classify amino acids based on their chemical classification, based on the metabolic classification, based on activity, and all those classification. And you will be expected to be familiar with how to go about the classification and what they do in health and diseases. So quickly, I'll go to, because we have just 20 minutes for this class, out of which we spend some minutes. We'll just start right away. And if you have any question, you can please call my attention to it by signifying using the icon on your Zoom. So thank you for being in this class. So the first classification that we're going to do is the chemical classification. And this is done according to the number of the carboxyl and the amino group that is available on the particular amino acid. And that is, in other words, that would be according to the net charge on the amino acid. So if we have a carboxyl, one carboxyl and two amino group, it shows that there will be a net amino group, one amino group, and that shows automatically the amino acid will be basic, because we know that NH2 from our previous classes, we said NH2 is basic. So it is what net charge is that determines the class of the amino acid. Only L-alpha amino acid occur in proteins. We said that in our previous class, and we'll continue to say that again. Only L-alpha amino acid occur in proteins. And under this chemical classification, we have the monobasic and we have monobasic. And here, what are we saying is either one is as one basic group or one carboxylic group. You know, at the end of the day, if you have one basic, one carboxylic, which is acidic, it becomes neutral, because the basic actually neutralizes the acidic. I hope we understand what we're talking about. So the basic actually neutralizes the acidic, and then you ultimately have a neutral or an uncharged amino acid. Example of those ones could be glycine, which we know to be the simplest amino acid. The next one also is alanine, you know, which has just a methyl group in addition to glycine. These are all neutral or uncharged amino acid, because they have just one basic and one acidic group, and each one will neutralize the other, leaving the amino acid to be neutral or uncharged. You know, those are the examples on the screen. You have the glycine, alanine, you have the branch amino acid, example of which is valine, leucine, isoleucine. You also have the neutral sulfur containing, which is the cysteine and the methionine. We know those are the sulfur containing. Cysteine is not involved in proteins, we know, because we know that cysteine is basically two molecules of cysteine coming together. I hope we still remember that. Is a dimer of cysteine linked by the sulfur to sulfur bond. You know, so I'll just continue from there. So another one is the neutral. I'm sorry, I move faster. Is a neutral hydroxyl amino acid also. And out of that, example of that is threonine, which is alcohol side group, is a target for phosphorylation of proteins. Another one is the neutral aromatic amino acid, phenylalanine, tyrosine, tryptophan. All of these are neutral amino acid. The next one is also neutral heterocyclic amino acid, and we said tryptophan is that. And we all know that tryptophan has an indole ring. Proline also has a ring, and that is why we said proline is actually not a direct amino acid, but an amino acid. Here we have the skeleton of so many of these amino acids. Some of them have the phosphoglycerate, those that are in the phosphoglycerate family have the phosphoglycerate skeleton. Those that have four carbon units have the oxaloacetate skeleton. An example of that is asparagine and aspartate. And those that have two carbon compounds are the pyruvate family. Example of that is alanine. And we have those that have those are 5 carbon compound within them. Those are the glutamates, glutamine, and proline. Those are in the same family. Also, we know that the second group is the basic amino acid. We said one are the neutral or the non-charged. The second group is the basic amino acid, and these contain 2 or more amino groups or nitrogen atoms that act as base, that is, they can bind proton. At physiological pH, basic amino acid will be positively charged because they have a net amino group, a net positive charge. An example of those are lysine, arginine, histidine, and all those. The third group is the acidic amino acid, and their amide groups, the acidic amino acid and the amide groups. Those are at physiological pH, they will carry negative charge. If you remember, we said the basic amino acid will carry positive charge at physiological pH. Example of acidic are the aspartic and the glutamic acid. We know aspartic is the 4 carbon amino acid, whereas glutamic is the 5 carbon. And the amide groups of those amino acid are asparagine and glutamine, respectively. And these are classified as neutral amino acid. We have this, there. So if we want to classify, we said classification based on chemical classification. The next form of classification is classification based on polarity of the side chain. Don't forget, in our earlier class, we said that amino acid has 4 basic units: the R group, the hydrogen group, the carboxyl group, and the amino group. Now we are saying when it is being classified based on polarity of the side chain, which is the R, you can have polar amino acid, you can have non-polar amino acid. So the first in that group where we're going to talk about this evening is the polar amino acid, where the R contains the polar hydrophilic group, so can form hydrogen bond with water. And the R, which is the side chain, may contain hydroxy group, may contain sulfhydric group, which is the SH group. We can also contain amide group. An example of those that contain the hydroxy group is serine, threonine, tyrosine, because of the R group that is hydroxyl in nature. And those that have the SH group or the sulfhydric group are the cysteine, and then those that have the amide group are the glutamine and asparagine. And we know that the amino group or the nitrogen, they act as a base. And when the R group, is in the carboxylic form, then we know carboxylic group is acidic in nature. So you're going to have acidic amino acid. You're going to have acidic because the polarity now is towards acidity. And then that shows the pH will be lower, whereas if it were to be basic, the pH will be above 7. An example of the acidic is the aspartic acid and the glutamic acid. And don't forget, we said the aspartic acid is the 4, which is the oxaloacetate family, whereas the glutamic acid is in the 5 carbon family. Here are the different amino acid. We have the hydrophobic here, we have the aromatic here, we have the amide here, different groups with their different structure. Here we have the aromatic, phenylalanine, tyrosine, tryptophan, the acidic, aspartic, glutamic, the amide group, the asparagine, glutamine, the basic group, which are the histidine, lysine, arginine, and I believe we still remember that song that helped us to classify them into different groups. So now, we're talking about the non-polar. We said the polar, now we're talking about the non-polar. Those that are polar are basically due to the, net charge, which is either positive or negative. When it is positive, we know it is basic, and when it is negative, we know it is acidic. So now, talking about the non-polar amino acid, here the R is alkyl hydrophobic group, which cannot enter in hydrogen bond formation. And 9 amino acids have been known to be non-polar. Example of those are glycine, alanine, valine, up till methionine. And having this GALT-VIMP is an acronym that can help us to memorize them. So the third nutritional classification is a third form of classification that helps us, to classify based on nutrition, based on how important they are to our health. So now that we have 3 of those, we have the essential, we have semi-essential, and we have the non-essential. Let's quickly take them one after the other. The essential or indispensable, just from the name, indispensable, are those that cannot be formed in the body and so has to be taken in diet. So I'm sure we have come about essential amino acid, essential fatty acid. So when anything is essential, it shows that they are very important, but they are not being produced by the body. So they are needed to be taken in, from the diet. And these are very important because they are required for proper growth and maintenance of the individual, animal, and man. And the deficiency will affect the growth, will affect health, will affect the protein synthesis. So the second group is the semi-essential amino acid. And these are formed in the body, but not in sufficient amount for body requirements, especially in children. They are formed in the body, but not sufficient. So not that the body does not form these, but they are not formed in sufficient amounts in the body, especially in children. Here are examples of essential amino acid: isoleucine, leucine, lysine, phenylalanine, choline, tryptophan. All these are examples of essential amino acid. So now talking about the essential, and we said essential, I've actually depicted them as Villa HM, 10,000 pounds, just for, ease of identification for me. You also can give an acronym that will help you to remember them. The third group are the non-essential amino acids, just from the name. These are the rest of the 20 standard amino acids that are formed in the body in amounts that are enough for adults and children. And these are the remaining 10 amino acid. The other classification is the metabolic classification. When we talk about the metabolic classification, we are talking about classification based on the metabolic products. And here you can have ketogenic, you can have glucogenic, you can actually have mixed, amino acid. That is, that has both ketogenic and glucogenic end products. Let's quickly take them one after the other before we round off this evening. Based on metabolic or degradation products of amino acids, and that shows that it will be ketogenic amino acid. These are amino acids that give ketone bodies, as metabolic end products. An example of those are lysine and leucine, and these are the only pure ketogenic amino acid. I believe that is a point that we need to take note of. They are the purely ketogenic amino acid. We also have the mixed ketogenic and glucogenic amino acid. These give both ketogenic bodies and glucose. An example of these are isoleucine, phenylalanine, tyrosine, and tryptophan. Those are the 4 mixed amino acids that we have. The third group are the glucogenic amino acid, and these are amino acids by catabolism yield products. They yield products that enter in glycogen and glucose formation, and these include the rest of the amino acid. I'll quickly talk about, few of this, and then we'll close for the day. When we talk about the glucogenic or ketogenic amino acid, what readily comes to mind is fasting condition. During fasting, those amino acids that are degraded to pyruvate or 4 or 5 carbon intermediates of the citric acid cycle can be used as substrate for gluconeogenesis. We all know that during fasting, there's no intake of food and the body still needs energy. So what happens? What happens is that the body will now tend to use up the fat and the amino acid. There's nothing called protein sparing. When there's food, there's protein sparing. But when there's no food, during fast, as in fasting, what you see is that the amino acids are degraded into pyruvate, and from pyruvate, they can be used up for energy process. And we know what gluconeogenesis is. Can somebody remind us what gluconeogenesis is? Gluconeogenesis, can somebody remind us what gluconeogenesis is? Can we have a volunteer, please? Gluconeogenesis, I believe in pathology also you must have treated gluconeogenesis, also in medicine. Gluconeogenesis, can nobody help us out just from the name?
Good afternoon.
Somebody to help us out, please.
Good afternoon.
Yes. I think you should signify first and then I will call you. You know how to do that? Just signify. Let me see your hand and then I'll call you. Let's see a virtual hand and then I'll call you. I believe we know how to do that. Yes or no? Yes, Ezekiel. I can see your hand. So you have the floor.
Yes, ma'am. Good afternoon, ma'am.
Good afternoon.
Gluconeogenesis is the biological process of synthesizing glucose from other sources like protein and
from non-carbohydrate sources. Thank you very much. Thank you very much. Thank you very much. Thank you. Thank you. Let's quickly go on. Thank you. So amino acids that are degraded to acetyl-CoA provide substrate for ketogenesis. We know that. And this distinction has led to the classification of amino acid as glucogenic or ketogenic. And don't forget, we said there are just 2 purely ketogenic amino acids, and those are leucine and lysine. Whereas those that are mixed amino acid are 4, isoleucine, phenylalanine, tryptophan, and tyrosine. And then we have the remaining all being glucogenic, I hope you guys are still with me.
Yes,
yes, ma'am. With me.
Yes, ma'am.
Thank you.
Yes, ma'am. Yes, ma'am.
Thank you. So looking at this in summary this afternoon, looking at what we have on our screen is basically the classification. And we said amino acid is composed of the alpha carboxyl group, alpha amino group, which is the amino group, the side chain, and then the hydrogen. And this hydrogen is what helps it in hydrogen bonding, and if we look through the chart, we'll see that the carboxyl group is actually deep. When it's deprotonated at physiological pH. And so you have the carboxyl group, which is CO minus. Also the amino group, when it is protonated at physiological pH, you have a net positive charge, which will have the NH3 plus, Also the side chain, we said the side chain can be composed of non-polar side chain. We can have uncharged, polar side chain. We can have acidic side chain. We can have basic side chain. For the non-polar side chain, that shows the R can actually be alanine, glycine, isoleucine, valine, all those. And we know that all those, when they come in, they are non-polar side chain. The uncharged polar side chain are the asparagine, which are the amide form of aspartic acid, cysteine, glutamine, serine, threonine, and tyrosine. Those are the uncharged polar side chain. And the aspartic and glutamic acid are the acidic side chain, whereas the basic side chain are basically arginine, histidine, and lysine. Also, we know that the hydrogen portion, can be in form of hydrogen, and that hydrogen can actually possesses a buffering capacity. possessive buffering capacity. In conclusion, in proteins, most alpha carboxyl and alpha amino group of the amino acids are combined in peptide bonds, and therefore these groups are not available for chemical reactions. Once they are, bound together in peptide bonds, they will not be available for chemical reaction. And thus the chemical nature of the side chain will now determine the role that the amino acid will play in a protein, in particular. And we said the side chain now determines, the function or the properties of the amino acid, how the protein folds into its nature, and its conformation is as a result of the net charge or the R group, on the amino acid. Ladies and gentlemen, on this note, we will end this class, and I would like to take our reactions and questions. For now, can we all put on our video as I take a shot of all of us? Maybe I should stop sharing first before I do that. Can somebody take a photo of this sharing before I unshare, please? Toby, can you do that for me?
Maybe a screenshot.
Yes, please take a screenshot of it. Thank you. Thank you very much. Have you done that?
Yes, ma'am.
Yes, so I'm going to stop sharing now, and we all will put on our video so that we can take the last. I want to see our beautiful announcer. Oh, look at Ezekiel, long time. So can somebody take this, a screenshot of all of us also here before I stop the recording? Oh, Ezekiel, you have a question? Sorry, please, can somebody take a screenshot of this before